Agriculture recreates the world's food supply every year, from seed, soil, sunlight, and water. Since the first cultivated crops, we have selected which plants to use based off their characteristics. Though the methods have changed, the goal remains the same. We use plant breeding to solve problems. My name is Kelly, and I'm going to walk you through how modern agriculture both discovers and creates solutions. This video is going to focus on how plant breeders identify disease resistance and incorporate this resistance into new varieties in order to minimize pesticide use and solve problems. Plant breeding is the science of crop improvement and works through identifying genetic variation and incorporating this variation through pollination and selection. We're going to focus on a disease problem with tomatoes known as bacterial spot. Plant loss to disease can be dramatic. In any given season, reported losses to tomatoes can be as high as 10 to 15%.
The impact of bacterial spot is, we estimated around 13 ton to the acre on an average crop. That cost you can take times the price of the tomatoes, which is usually around $1,300 per acre. There's 20,000 acres in the Midwest of tomatoes, so you're talking a pretty substantial decrease in income.
Tell us how you first learned of the new bacterial spot problem.
Well, we got telephone calls from growers in the region, and they had a very severe problem on tomatoes and asked us to come and look. It's something they hadn't seen before. The symptoms were a bit different from the normal bacterial spot that we see. So I went out there, collected a lot of samples from 20 or 30 different fields, and looked at that issue, brought them back to the lab, and diagnosed the problem. It took some time to do that, but the symptoms in the field were different. There wasn't as much foliar disease as we usually see, and the really interesting thing was that there are very large spots on the fruits, which are quite different than bacterial spot, which is a small, couple millimeter size spot, but these were probably almost a centimeter, and they ruptured the skin so that you had sort of a star-shaped pattern. So that was quite different than what we'd seen in the past. And it didn't seem, there could be some differences by variety, hard to tell in that situation just by going through the fields, but it was definitely a very widespread problem.
Plant breeders look for new traits, including resistance, in diverse collections of plant material which we call germplasm. These resources may be cultivated types, old land races, or even related wild species. The center of origin for tomato is Andean South America, where great diversity exists. Much of this variation is housed in germplasm collections, such as the United States Department of Agriculture's National Plant Germplasm System and the Tomato Genetics Resource Center. Seed resources are distributed to public and private plant breeders free of charge. Collections of diverse plant material are then screened for resistance. These screens may occur in the greenhouse or field. The bacterial spot pathogen, Xanthomonas, is cultured and used to inoculate plants. Typically, plants are inoculated in the greenhouse by injecting the bacteria directly into a leaf. Inoculations for field experiments are done by spraying a solution of the bacteria over plants prior to transplant. Within a short period of time, often just 1 or 2 days, resistant plants will have small dark spots. These dark spots indicate that the plants have contained the infection, whereas the susceptible plants will show signs of a rampant and uncontrolled infection. Eventually, susceptible plants may be completely overcome by disease and die. Once a source of resistance is identified, the plant breeder begins to cross-pollinate to combine resistance with other desirable traits. This process leads to development of populations that can be used for selection, which is the process of eliminating undesirable plants and keeping plants with the combination of desired traits. These populations can also be used to learn about the inheritance of resistance.
We look for a few things in a variety. Probably the most is going to be its ruggedness or its durability, and that's really in essence to how it's going to withstand the climactic impact. And then second of all is the marketability of the fruit coming off of it. It has to be a good clean fruit, free of any blemishes, black marks, spot, speck.
Eventually, new varieties with the resistance may be available to the public. Because new problems continually arise, plant breeders are always working to develop varieties to overcome the challenges presented.